Buenas noches, ¿cómo están? Bueno, muchas gracias a todas las personas que están aquí presentes. Gracias a las que están aquí desde hace muchas horas también, porque como yo empezamos hoy la jornada bastante temprano, entonces les voy a hacer una presentación. Hay algunos que estuvieron desde de la mañana, pero hay varias personas que no, entonces voy a presentar a nuestro invitado del día de hoy. Para mí es un gusto recibir aquí en la Universidad de Costa Rica al profesor Manuel Spitschan, que es una de las voces referente a nivel internacional en el tema de cronobiología y el estudio de la exposición a la luz y los efectos sobre el sueño, los ritmos circadianos y la salud. Manuel desarrolla su trabajo en entre la Universidad Técnica de Múnich y el Max Planck Instituto de Biología Cibernética. Y creo que este es un contexto muy valioso para este tipo de investigaciones, ya que en el trópico tenemos iluminación bastante abundante. Sin embargo, creo que no siempre estamos tomando pues bastante provecho de esta condición. La charla de hoy, como ven, se llama Viviendo con el Sol Tropical, la luz, el sueño y la salud circadiana. Y esto es parte de una visita académica que están realizando el doctor Johannes Sauerner, que está aquí presente, y el profesor Manuel Spitschan a Costa Rica en el marco de una visita académica y de investigación. Entonces, sin más, les damos la bienvenida a Manuel. Muchas gracias, Manuel.
Muchas gracias, Andrea, por invitarme. Es mi primera vez en Costa Rica. Me ha encantado, estoy muy encantado de estar aquí. Pero ahora paso al inglés. Es más fácil por mí. So I'll talk about research that we've been doing in the topic of light exposure, sleep and circadian health. And I want to really give you an overview of how the field of sleep science, chronobiology, and more generally biomedical science thinks about circadian rhythms, our biological clock, and so on. To get us started, I would like to ask you a few questions. And what I'd like you to do is raise your hand if you say yes to these questions. The first one is, do you use the night shift mode on your phone in the evening? This is the one that dims it or makes it more orange. There's a few people who must have disabled it or don't have a phone. Do you dim your environment from 3 hours before going to bed? That's 2, 3 illuminated people. Do you spend most of your day in a well-illuminated office or classroom, university classroom? A lot more. And do you spend as much time in daylight as possible? So this is, I think, also very low responders. So what I want to walk you through today is why these questions matter for our health, for our circadian rhythms, and for our sleep. And hopefully by the end of this talk, this presentation, you'll leave with the sense of why light exposure is very important for us. And I'm very excited to be here because most of the research that my team has been doing and that the field has been doing really is done in the northern hemisphere. And as a consequence, I think we lack a lot of understanding in terms of how light exposure affects human physiology, human behavior in equatorial settings like Costa Rica. I want to give you the message that I want to leave you with before I start the talk. So if there's anything you remember from this talk, I hope it's this: bright days and dark nights. And I'll expand on why this is important throughout the next hour or so. I have prepared basically 4 topic areas that hopefully will be of interest to you. We'll talk about how the eye talks to the brain. We'll talk about exposure to light and what it does with our circadian clock and our sleep cycle. We'll talk about what types of exposures we might be exposed to in terms of light, and also how different people might respond to light differently. And I think this is particularly interesting, not just from a fundamental point of view, but also in terms of how we think about how we should use light in the built environment. Now, I'm based in Tübingen and Munich, which is in Germany. Here's two pictures that give you a little bit of a flavor of the cityscape and the landscape that we're in. Munich is a very big city. Tübingen is a small university town. And it's been really nice to be there for the last 4.5 years, to develop my research group. And I just want to give you a quick insight into where my research group is based. We're set up in Munich and Tübingen, but also in Singapore, which of course is a lot more equatorial than Germany. And we're trying to really integrate these types of research questions in a coherent sort of study program. If you're interested in learning more about the work we do, this is our lab website. And then, of course, also we do spend a lot of time in the lab, but we also spend time outside of the lab. And so most recently, we spent a good amount of time on the biggest mountain of Germany to discuss how we want to do science and kind of calibrate our expectations for what's important. So we go back to this 4 topic areas, and I want to start really with this fundamental question of how the eye talks to the brain. And so all of you probably learned a lot of things about the eye and the retina and the sensors in the eye that help us see during the day and during the night. And really there has been a lot of development in how we understand the fundamental biology of vision. So vision, sensing, seeing the environment, and particularly with respect to the effects of light on our physiology, we really need to rethink what it means for us to be in a bright or dim environment. You've probably seen a diagram of this, maybe for some of you a long time ago. We have light that enters the eye through the lens. The lens is sort of a semi-opaque body that, of course, changes with age. You have a lot more a yellow lens when you're older. Then the light reaches the back of the eye. And there's these photoreceptor cells that are waiting to pick up the light and convert it into signals that are useful for our brain. And the 2 photoreceptor types that we know or have known for about 200 years are the rods and the cones. There's 3 types of cones that help us see color, help us see vision, help us see spatial detail, motion, and really underlie everything that we usually associate with visual perception, right? So right now, as you're reading this, as you're seeing the orange of this eye, all of this is done by the cones. There's another set of cells that are called the rods. The rods are not existent in the center of our vision, and they are sensitive in dim conditions. So when it gets night and there's no light outside, we might still see some shapes, and that's because the rods are there to support this visual function that is quite rudimentary. And in addition to these rods and the cones, there's another class of cells, and these are called, scientists like to make up long and complicated names and then abbreviations. These are called the intrinsically photosensitive retinal ganglion cells. We abbreviate these as IPRGCs. The way you need to think about the retina is really it collects light from the outside world. The sensors are activated and kind of form an image that is sent to the brain. And this sort of sensory processing happens because there are cells that take the information and combine it and send it onwards outside to leave the eye and then go to the brain. And some of these cells that are sort of, we call them relay stations, are what we call intrinsically photosensitive. So they are themselves responding to light. The consequence of this is if you don't have rods or you don't have cones, there are still cells in your eye that pick up how bright it is in the environment. And this is very important because there are people who are blind, they don't have functioning cones and functioning rods, who if you go to an ophthalmologist would be blind, visually blind, who still have a response to light. And that is, 1, their pupil size will change if they're exposed to light, albeit very slow. And 2, when they're exposed to bright light, their melatonin will be suppressed. Melatonin, I will talk about in a second, is a hormone that's produced by the body. Some of you might already be in the rising phase of melatonin, depending on what your chronotype is. And in blind people, if you expose them to bright light, you can reduce the production of this hormone melatonin, even though they are insensitive to the visual environment. So this tells us something that's fundamental here, which is we have a pathway that underlies vision, and we have a pathway that we are not consciously aware of that is nonetheless light sensitive, and that is mediated by these IPRGCs. So the cones allow us to see color, motion, spatial detail. The rods allow us to see rudimentary spatial detail under dim light, and the IPRGCs convert light into signals that influence many physiological functions. If you were here this morning, you have seen this diagram before. We have, of course, we're talking about the brain. The brain is as the central sort of conductor of everything that happens in physiology. The retinal contains these photoreceptors and then project via the optic nerve to different parts of the brain. And when we're talking about vision, so color, motion, spatial detail, these cells go or send their signals to the back of the brain. The back of the brain is fundamentally important. When you have a lesion there because of stroke or some other damage, you're losing your vision. And fundamentally, this is really the pathway that subserves everything that's related to our vision and visual function. And this other pathway that we don't know much about yet, that we're trying to research, is called, and this is again a long name, the retinal hypothalamic pathway, connecting the eye to a small structure roughly the size of a grain of rice that is super important for the entrainment of our biological clock. So the way you think about this, light in the environment activates the cells in the eye, then sends a signal to the SCN, it's the structure in the brain, and there it is responsible for modulating how much melatonin is produced. Some of you might be aware of melatonin. You can buy it as an over-the-counter drug. I don't know what the legislation is here in Costa Rica. We can buy it in a drug, it's a you can get it over the counter as gummy bears, right? So this is a hormone that our body naturally produces. It's not produced during the day, and it starts in production a few hours before your habitual bedtime, so before you usually go to bed. And some people, of course, use it to induce sleep or shorten how long it takes to fall asleep, or if you're traveling across time zones, it can help reset your biological clock. Yeah. And then the structure, the SCN, basically all organ systems and many physiological functions are undergoing 24-hour rhythms. So this includes our cardiovascular system, this includes immune function, and also metabolism. Right? So how our body responds to food intake, meals, will be very much depending on when you are eating it. Right? I think everyone has probably felt they've eaten at the wrong time, and sometimes the metabolism just is slower just because you are at an inopportune, a disadvantageous time of your circadian clock. For us, so this is, I mentioned this earlier, so melatonin starts in production. This is for a person, an example. It starts in production, then it's sort of plateauing at night and then goes down again. And so this happens also when you take melatonin as a pill or as a gummy bear, but we all have this natural rhythm of melatonin. No melatonin is produced during the day, and you have melatonin that's produced in the evening and then produced throughout the night. When you wake up, you still might have a little bit of melatonin, but it really drops, a few hours before you usually go to bed. There's a lot of variability in how much melatonin we produce, but for us, this is really our main way that we can determine what is someone's biological clock doing at any given time. And we can measure melatonin through multiple ways. The most convenient way that we use is saliva. So we take saliva samples and then later on do the biochemical assays based on little cotton swabs that store up to 1 milliliter of saliva or so. Of course, I talked about the eye, I talked about the circadian pacemaker. Ultimately, what matters is how much light reaches the back of the eye, the retina. And that is controlled by many different things, of course. It depends on the built environment, how much light there is, where you're looking, where your body is turned to, head movements, eye movements. But we also know that it depends on how big your pupil size is. if we're here in Johannes' talk earlier today, he mentioned this, and I just want to give you an insight of what this looks like. This is one of my favorite videos that I've ever produced in my scientific career. And I'd like to think that if my scientific career at some point comes to an end, I have a second chance as a horror film director. So these are 64 eyes, I think, in 64 individuals. So interestingly, usually this splits the room in half. There's people who find this absolutely fascinating and want to keep looking at it, and people who just find this absolutely disgusting. And so basically what you see here, these are individuals that undergo a procedure in our laboratory, and what you see, they're all in the same conditions, they're all in the same lighting. Some of them have smaller pupils and some of them have bigger pupils. So for example, compare these two individuals with relatively big pupils to, say, this individual here. And so already as early as how much light goes into your eye, there's enormous individual variability, because think about this as the aperture of a camera, right? The bigger it is, the more light gets in, the smaller it is, the less light gets in, and there's a certain amount of modulation then of how much light gets in depending on the size of the pupil. You want to see it again? Oh, no. So, how the eye talks to the brain, light detection beyond vision. We know that visual function is really important, but there's another pathway that's sort of unconscious that is important for how our biological clock is entrained, when we sleep, how we sleep, production of hormones, and is really a fundamental pathway that we need to understand scientifically, because it controls almost all aspects of our 24-hour physiology and behavior. So moving on from the retina and the front end of the system, what I'd like to talk about is what happens when we are exposed to light at different times of day, and what that means for our biological clock. Now, I've said the word biological clock quite often. I want to give you a little bit of a feel for what we mean when we say biological clock. And for that, we look at one of the founders of modern chronobiology, a French biomedical scientist and cave explorer called Michel Siffre, who passed away, I think, last year after a long career exploring caves and exploring the biological clock. And so Michel Siffre in the 1950s and 1960s did studies where he spent long periods of time in a cave. Why in a cave? Of course, in a cave, you don't have daylight. And so what he wanted to test was the question, or asked what he wanted to test was, what happens when you spend your days in time-free conditions? So we remove this light-dark cycle that is given by daylight and see what happens to our biological clock, what happens to our sleep-wake cycle. Of course, he took a lot of diary. There's, of course, some EEG measurements that he did, but basically, and he took some philosophy as well, basically spending some good amount of time there and then asking, well, what happens with my daily sleep-wake cycles? Did that come out of the cave, that animal? No. So here's what happened to Michel Siffre. So I want to orient you to these plots. If you haven't never seen these plots, they're a little bit hard to understand. On the x-axis, we have time of day, and we start with 8 a.m. on the first day, go up to midnight, then this is the second day, and then this is the third day. So on the x-axis, we always have roughly 72 hours. Here it's 64 hours because we start at 8 a.m. On the y-axis, we have the day that Michel Siffre spent in the cave. And so he goes in the cave on day 1, spends day 2, calendar days, right? So say 21st of March, 22nd of March, 23rd of March, and so on. And then he leaves at some point around day 60. In light gray, this is showing when he was awake. In dark gray, this is showing when he was asleep. And so what you can see here is if you plot it as a function of local time and local calendar day, over time, when he goes to bed and when he wakes up, drifts, right? So if you were always going to bed at the same time, and if you're always waking up at the same time, you would have a straight line that goes like this, maybe a little bit of wobble, because of course, maybe sometimes you sleep a little bit longer, a little bit shorter. But under these conditions, when Michel Cifre was without daylight, basically the biological clock was still running, but was running not with a period of 24 hours, but a little bit longer. So if it was 24 hours, it would be straight down. If it's longer, it would be shifted or tilted like this. If it was shorter, it would look like this, so it'd go on the other side. And so what this tells us is when you remove the light-dark cycle, the biological clock keeps ticking, but it is very imprecise. So these studies have been repeated. One example of these are the famous bunker experiments in the south of Munich, where also a researcher ran similar experiments. Basically, people were under controlled conditions for a while, and then on day 9 or day 8, entered this protocol where basically they were able to self-select when they would turn the light on, when they would turn the light off, but there was no natural 24-hour rhythm. Right? So imagine we shut the blinds, no light coming in, and you just live however you want. When you wake up, you turn the light on. When you go to bed, you turn the light off. And this is similarly, you see this drift in the timing of the clock. One of my favorite anecdotes of science is a quote from this paper that described the experiments, which I'll read to you. We supply the subject with fresh food and other necessities, such as 1 bottle of Andex beer daily. So Andex is the town where these experiments took place. And so in the 60s, you could still pay the participants with beer. Nowadays, impossible. We would not get this approved by the ethics committee. These experiments were done in Bavaria, so this explains a little bit why you'd get a beer every day. Now, you'd think, oh, this is sort of early science, right, 1960s, 1970s. I'm happy to report that there's still folks who spend time in caves for long periods of time to understand a little bit about the biological clock. There's a project a few years ago in France, something about French caves that makes you want to spend time with them. Deep Time Project investigated how a lack of external contact with the sense of time, and 2/3 wanted to stay longer. I will say, I think the most impressive study that I've come across, or the most impressive event that I came across, was a woman in Spain who emerges after spending 500 days living alone in a cave. She says she endured swarms of flies, read 60 books, and never missed the sunlight. So what's interesting, I think, here is that, of course, these are studies that are kind of very similar to the types of studies that we would run in chronobiology. And, of course, most of these folks are probably cave explorers and are able to combine, right, a hobby with something that's interesting. I love this quote. She had an odd, tantalizing vision of roast chicken. So at some point, I think if you spend enough time in time-free conditions, you fantasize about eating roast chicken. There's one important aspect here, which is, so 500 days is a long time. There's more than a year and a half, roughly. She reported she was able to track time, and not by counting the days, because, of course, if the days are shifting like this, you don't really have anything to work with. But there's another biological timing system that around half of people in the audience will know, and that is the menstrual cycle. So she was able to track the number of cycles she was undergoing, and so roughly was estimating how many days she was in that cave. The biological clock is really fundamental to many different things here. This is an odd clock because it shows 24 hours. We start in the morning. Usually when we wake up, the hormone cortisol is secreted. Cortisol is a hormone that's sometimes called the stress hormone. When you're under very stressful conditions, like an exam, or you're at the airport and you forgot your passport, you have a cortisol spike. It usually helps us get going during the day and sort of kick our day off. We have then, of course, whenever we have our first meal, so whenever we have breakfast, the first secretion of insulin. Early in the morning, peak in alertness, and then, interestingly, sort of towards the evening, greatest cardiovascular and skeletal muscle strength. So if anyone here is an athlete or likes to do weightlifting or whatever, usually you have a peak in the late afternoon. Then you have a peak in body temperature and blood pressure, and then, as I mentioned earlier, around the evening, melatonin secretion begins. So around 1 to 3 hours before you usually go to bed. Then you sleep. You have your, what we call the mid-sleep, of course, sometime between 2 to 4 a.m., and then the cycle starts again. And so what this shows is there's many different functions in our physiology, whether it's muscular function, whether it's blood pressure, whether it's hormone production, whether it's cognition, right, that is driven by this 24-hour rhythm. And so there's very interesting studies that, for example, show that our decision-making ability depends, of course, on the time of day. If you've been sleep deprived, I think we all know this, but also there are these interesting this large-scale studies where they investigated whether exam performance in students depends on whether the exam is held at 8, 9, or 10 a.m. And so you see these effects basically in all, if you look close enough, you see them really in all areas of human cognition, human life, and so on. One of them being athletic performance. So this is a study that came out based on a sort of data that's out there, right? So you can look at finishing times in swimmers and Olympic swimmers, in the heats, in the semifinals, and the finals, and you can then plot it as a function of time of day, right? These are public data. You can easily do this. And what you can nicely see here, so this is sort of the individual data, this is the average data. You can nicely see that you have these variations, and what you find here is basically the fastest time will be sometime around 5 p.m. So again, I think this is the same story that we have here. You have the greatest strength sometime in the late afternoon, and you can see it even in these data that were never planned to be collected to test anything about human biology, right? You can just get this from the internet where you get all the times. And so what we call circadian variation, circadian, approximately 24-hour, we see that in really many, many areas of life. And for us, of course, then as chronobiologists, we're quite interested in what do we do with this? Exposure to light at different times of day changes when we sleep and when we're awake. And light, the light-dark cycle is really the main signal that ensures that our circadian system is entrained to the light environment that's given by daylight, right? So the Earth's rotation changes how much light there is available, and of course during the night there's none. We also have the ability to extend the day, of course, with artificial light. So right now the sun has set, yet we have a good amount of photons reaching our eyes right now, right? So the light-dark cycle given by the natural environment, but also by just generally any light that we are exposed to. Light directly influences the biological clock in the brain, regulates sleep-wake cycles and other physiological rhythms. And so now this is very important. Light in the morning can move the circadian clock earlier. Light in the evening can move it later. So we call this advancing second animal. We are probably disturbing them right now because of the light here, right? So they think it's day. So light in the morning can advance the circadian clock, light in the evening can delay the circadian clock, right? So if you have bright exposure, you might go to bed earlier, right? If you have bright exposure in the morning, if you have bright exposure in the evening, you can shift it. And there's another effect that's really important. The brighter the exposure is that you have during the day, the stronger your circadian system is entrained, and that has consequences, for example, how deep we sleep. So the exposures that you get throughout the day are fundamentally important for your sleep quality. This is a cartoon that I pulled from the New Yorker. It's from 2022. I don't know if anyone close to you looks at your phone late at night. This is a common thing. I love this quote: "Careful, they say that blue light and the world falling apart can affect your sleep." Every year I look at this and I say, this cannot be any truer. So, exposure to light at different times of day alters the timing of sleep wakefulness. And now I want to really go into this question of what do we mean with light? What is light? I think we all have an intuitive understanding of what light is. If you're an architect, I think you use it in design, in evaluating spaces. And for us as scientists, it's of course interesting to understand what is light for you, right? What is the priority that you give light? How important is it? We have two animals on the ground that have dropped. So, type of light that we see throughout the day, daylight versus electric light. And I want to start with sort of enumerating, listing the types of light that you might be exposed to at any given point during the day. So, this is someone sitting on the couch, just looking at their phone, but at the same time, of course, there's daylight coming in through the window. So we have light source 1, light source 2 is daylight. We have maybe overhead lighting. This is maybe an old incandescent light. It's a little bit orange. Then we have this task lamp or sort of decoration lamp that comes in with a little bit of more orange light. And then this person also put a Netflix show on, but forgot that it was running because there was something more interesting to look at on the phone. And so all of this combines and leads to what we call light exposure. So all these sources at any given point of time will be visible to you and will affect your circadian clock. And so the interesting thing here is, of course, this person will be moving their head, will be moving their eyes. And so at any given moment in time, really there's a lot of dynamic variation how much light they get. So right now, as you're looking through the room and maybe looking at this side or whatever, or looking at the other animals that are in the room, you will find that the light exposure is quite dynamic, quite variable, and quite noisy in a way. And of course, one thing that's important, this person sits in a specific position on the sofa, but there's another person in the room, and this person gets a completely different light exposure because, of course, they're facing away from the laptop. The window is a little bit off, not looking at this light exposure. And so what this means is that whoever is in a given room will experience a different version of the space, right? I think this is pretty clear, but it means for us that we basically really need to measure what exposure this person is getting, this kid, and what exposure this person is getting. By the way, I think this is a highly unrealistic picture. As a father of an 8-month-old, once there is a screen, they will look at it. So I think we need to maybe turn them around by 180 degrees. Now, we have ways that we can talk about light, and this really comes from physics, right? So physicists know how to characterize and measure light, and so now I already gave you maybe a traumatic flashback of high school biology. I'll give you another traumatic flashback to high school physics. So light is described by how much energy there is at each wavelength across the visible spectrum. And so here I'm showing you different types of spectra, different types of light sources that contain different amount of photons at different wavelengths. So wavelengths here on the x-axis. And so for example, when you're looking at daylight, there's never one daylight. There's different types of daylight. It depends on whether the sky is overcast, the position of the sun. All of this will change what we call the spectral composition of daylight. Now, we have different light sources. We have artificial light sources such as LEDs, fluorescent lamps, and we also have sort of this is what's called standard illuminant, but really this is an incandescent lamp. All of these have roughly the same color temperature, so they all have sort of an orange glow. They all have the same intensity, but they have very different spectra. And this is ultimately because we only have 3 different types of cones that help us see intensity and color. And so if you're thinking about trying to find, so this is a mathematical problem, there's many different spectra, right, that when you project them to a 3-dimensional space, occupy the same points. It's also the reason why we can reproduce colors through printing by mixing paints, inks. It's also the reason why we can almost approximate any color with projectors. It's because we only have 3 photoreceptors, 3 cone classes, and so you only need 3 lamps that you mix in different proportions to achieve one intensity and color combination. This means that these spectra will look the same to us, in principle, but will have very different effects on how they affect our physiology. Daylight has what we call a broad spectrum, right? So when we're thinking about what is the natural stimulus to the circadian system or to many aspects of biology, this is what organisms evolved under, and it has really power at many different wavelengths. There's one interesting side note here, which is, well, you know, so the sun is a radiator that just produces radiation. But of course, we are never exposed to the sun directly because light is filtered through the atmosphere. And the reason why you have these different notches here, these little bumps and grooves, is because the atmosphere filters light depending on the composition of the atmosphere. So sunlight comes from the sun, the atmosphere filters it, and that's why we have fewer photons, for example, at this wavelength, because that's the absorption of a specific molecule in the atmosphere. As a consequence, you can actually learn a lot about the atmosphere by looking at what's the radiation that's arriving on the Earth's surface. Sometimes you have these, so this, I don't know if this is a common occurrence here, but sometimes we have, of course not, maybe similar events exist here, but we sometimes get sand from Sahara in Northern Europe. And when you do this, of course, the daylight is completely filtered. It's a very strange appearance because you're never exposed to this type of environmental illumination. This is a data set, very data dense, but I just want to walk you through something that I think is fundamentally important for how we think about light in the built environment outdoors. So, of course, we can build spaces that are illuminated in specific ways. We can spec their color temperature, we can spec the illuminance, but of course, we also need to think about not just what do we do in inside spaces, but also how does light propagate in the general environment. And of course, we have not just us in the environment, but all kinds of fauna and flora that are affected by light. And so a few years ago, while I was doing my PhD, I was interested in this topic of light pollution. So light pollution refers to emissions of light that make us not able to see the night sky and the stars anymore. This is a topic that's particularly interesting to astronomers because they want to see the night sky, and if there's glow of the sky because of light that just gets bounced back, they cannot observe the constellations, right? At the same time, light pollution also confuses lots of animals. So we know moths, for example, are confused by just lamps, right? We see them just dancing around lamps, even though they should not be there. And so what I did, I went to a part of Pennsylvania in the US that was free of light pollution. This is a very rural area. It used to be sort of lumber country, right? So we do get a lot of lumber extraction. I think extremely low build-up spaces, so no street lamps and so on. Very spooky at the time, but also very interesting because there was just no light pollution. You could see the Milky Way, basically. So I went there with a spectrometer and I measured basically these types of things over the course of the sun setting. So it turns out throughout the day, you know, these are the daylight spectra. Now, when the sun sets, so we have 3 phases of twilight: civil, nautical, and astronomical twilight. What you see is a short wavelength boost in energy. Of course, the light intensity gets a lot lower, but at the same time, you have more relative short wavelength light. The reason for this is atmospheric optics. You just have different types of scattering depending on the angle of the sun relative to the atmosphere. And so this causes the night sky or the twilight sky to be extremely blue. If you're a photographer, if you work with visual media, you might have come across the blue hour. You know about the blue hour, which is basically this short wavelength boost of light sometime around twilight. Some people think that twilight looks more orange. This is true when you look at the sun before it sets, right? But when the sun has disappeared below the horizon, it's really extremely short wavelength enhanced. And of course, in places close to the equator, twilight is a very short period. In Germany, twilight can last a long time, or if you go northern as well. Now, this is in the place where there was no light pollution. This is in the city of Philadelphia, highly built up, lots of industry, lots of street lamps. And what's interesting here is the day looks the same, The twilight, civil twilight, looks the same. And then at some point, it also looks the same, but what this shows is basically the emission of high-pressure sodium lamps. So this is street lighting that is shining up to the sky and then reflected back. So what this means is that in the city, it's very rare to see this natural twilight sequence. It's completely buried in the light pollution that you have from the light in the outside built environment. I mentioned earlier that light exposure is noisy, and I showed this example earlier today if you were at the other talk, but I just want to walk you through it again. We now have recommendations that tell us how much light exposure we should get, and this is really the work of many different individuals that have put together what we call a dose-response curve. So the brighter the light, the stronger the response. If you want to suppress your melatonin, the brighter the light, the more suppression you have. If you want to have a cognitive alerting effect of light at night, the stronger the effect, the brighter the light is. So it's kind of what you would expect. More means more, right? More light means, or the brighter the light, the stronger the response. I mentioned that we have different light exposures, and in very few cases we kind of have these hypothetical light exposures. So here I'm showing you 24 hours, I'm showing you the light intensity. Very few people stay at the same light exposure and then it goes down when you go to bed. Of course, when you're looking at different direction, if you're looking at daylight, this is what you might expect if you were collecting, or if you are stationary somewhere, just looking for daylight from above. And what you can see here, so this is evidently not from Costa Rica. You have the sun rising at 6 and the sun going down at 9. This is data from Germany. But what's really interesting is when you now measure in an individual the light exposure that they actually receive throughout the day. One thing that you can see is it's in many cases unrelated to when daylight starts, right? Very few people rise with the sunrise. Maybe it differs a little bit on where you are. Many people have alarm clocks, right? So they have blinds or curtains that leaves the light out. And then at some point they might wake up. The other feature that you can see here is that the exposure is very variable, but at the same time quite structured. So if I was to guess what this individual was doing throughout the day, they got up, then at some point between 11:45 and maybe 12:30 they had lunch outside. At some point they left maybe work and went to a park to spend some time outside, then went home and spent basically this period, which is after the sun has set, in electric lighting or in light exposure from phones or screens. Right? And we can measure this. We have devices that allow us to track light exposure. My colleague Johannes is wearing one right now, and I think he has been wearing it for quite a while. And so we just measure what the light exposure is that we get to derive these curves and try to understand what's the variability of how much light we're getting. And then we can do this a little exercise, which is we can ask, based on these recommendations, are we getting the right amount of light at the right time? And the answer is, in many cases, no. So this is where, of course, we're interested in thinking about how can we improve this, and part of it lies in the built environment, how we use light and spaces. Part of it is also our behavior, right? If I can choose to have my lunch outside or sit in front of my desk, I will have more light exposure if I spend it outside. So it's these little behaviors, little simple things that might change how much light you get. And this is where we've embarked on sort of trying to understand the light exposure across the globe. So the pattern of one's light exposure across the day and the year can be quite complex. It depends very much on where one is and what one does. And so we consider basically the exposure we get as determined by many different things: the location, the culture, the built environment, and then also behavior. And these are, of course, not independent factors. They are somewhat overlapping and drive ultimately what we get in terms of light exposure. And so this is sort of one example of how different behaviors are, how different architecture can be across different sites on Earth, what priorities there are. I should note, I think this is a very unrepresentative picture of London, if anyone has ever been to London. But so of course you do see quite complex seasonal dependencies, right? So of course you spend a lot of time in the park when it's warm, but you wouldn't do this in winter. And this very much depends on where you are, because seasons are depending on latitude. And so I want to walk you through just a couple of the quantitative data that we, including Johannes and many other teams, as well Andrea, have collected. I'll start with giving you a insight into the first data set that we collected of this sort. This was a master's thesis by now a PhD student. And the language of these plots is always going to be the same. You have here the location. You have here sort of an overview of when in the year the data were collected. Here, this shows the photo period. So photo period is the day length. How many hours of daylight are there? And how much did we sample in that specific data set? And then these types of plots that we've already seen, where we have 24 hours and then another 24 hours. And so in this specific data set, here are the recommendations. We want to be above this dashed line in most cases during the day. We want to be below this dashed line in the evening, and we want to be below this line in the sleep environment. Now, most people sleep in the dark or in dim conditions, so I think this is something that usually we are not too concerned with. Of course, light pollution is one issue. There's also the question whether 1 lux is not bright enough. If you put yourself in a 1 lux room, after a while it seems very bright. And so what you can see here, though, is that they never reach, on average, this threshold of what the recommendations should tell us. So this means we're not getting enough daylight, on average. Some people do, right? So these are statistical distributions. There will be some people who just have very bright daytime exposure. Some people might have bright days. But on average, in the population, or in the group that we sample, we don't see them reaching the threshold. For the evening, it's a little bit different. I think most people are in conditions where it is lower than the threshold, but of course this depends really on case-by-case basis. Munich, this is a different data set about a year later, only collected in summer, so very long days, so up to 16-hour long days. And here you can see that effectively the days are quite long, not only in how much daylight there is, but also in how much light exposure they get. This is in Madrid. So a little bit further south. This was collected in the winter months, right? So October to February. Photo period is a lot shorter compared to what we have in Germany, right? So this is 16 hours. Here it's only 10 hours to 12 hours. Yet the average light intensity is roughly the same, whether you're in Madrid in winter or whether you're in Munich in the summer. So what's interesting here is, of course, different conditions can give similar patterns, but what really is different here is the light exposure that these folks got, again, is below the threshold. And so in most data sets that we've looked at, we see this sort of relatively constant daytime light exposure that's below the recommended level, and then an evening light level that is quite constant as well because of artificial illumination. Now let's look at San Jose. So this is a data set that Andrea and her team collected, the largest data set in this whole campaign. So we are very close to the equator, right? 9.9 degrees north from the equator, spanning, of course, roughly on average 13 hours. There's not a lot of variation in the photo period because, of course, we're close to the equator. And so this is a very nicely sampled data set. And again, you see a similar pattern here. You see daytime light exposure that is too low, and a sort of second hump that corresponds to evening light exposure. I'm just going to switch between Madrid and San Jose. Right, so here the photo periods are very different. Right, so this is 10 hours. This is 13 hours roughly. And so overall, I think you see a very interesting pattern here, which is, of course, the photo period is longer. So the daytime light exposure is also overall longer. And so these types of data are incredibly interesting because, of course, we can now ask the question, what drives these differences in light exposure? Is it differences in how much light there is, differences in behavior, differences in the built environment, differences in the populations, differences in how much you seek light, right? And so this is, of course, something that we're quite interested in teasing apart, because when we're looking at the recommendations that I showed you earlier, This is really based on relatively homogeneous samples, mostly collected in Western Europe and North America, where, of course, when we're thinking about how light should be used, we really need to take a much more global stance and say, well, this needs to be expanded to more representative globally populations. I had the word tropical sun in the title, and of course, I wanted to kind of expand a little bit on my thoughts of why this is particularly interesting close to the equator and particularly interesting to look at in Costa Rica. Of course, daylight is intense. This is given by, the position of the sun and the ultimately lack of variability across the year. Day length is stable across the year, and then ultimately there's many different things that shape the real world light exposure. We did a wonderful tour of campus led by Jose and by Luis yesterday, where I noted that there's a lot of aspects of the built environment that really try to do one of two things. One is to manage rain and humidity and moisture, and the other thing is to manage heat from solar radiation. And so, of course, for us as people who are exposed to light, these factors really change how much exposure we get. And so this then leads us to this interesting paradox, which I would hopefully in the next couple of years try to resolve and try to understand, which is there's abundant light outdoors because of daylight, and yet many people spend much of the day indoors. And so the question isn't how much daylight or how much sun is available, but how much daylight reaches the eyes and when, and how do we square this up with general effects of light exposure? Right? So if we are avoiding daylight because of heat, or we avoiding the outside because of rain, how can we nonetheless still have a good signal to the circadian clock that entrains us? So I want to move on to my final section, and that is one, again, where we have the least amount of data, but I think the most interesting questions to ask, and that is same light, different effects. How do age, gender, and other factors influence the effects of light and influence the exposure that we might get? So we know higher light levels in the evening can increase the time to fall asleep. Higher light levels during the daytime can improve mood, and higher light levels during daytime can improve sleep quality the following day. So these are sort of fundamental knowledge now. We have established this through many different studies. But you notice here that this is relatively unspecific, right? What does it mean, higher light levels? What is the number? And what does that mean for a given individual? Of course, in biomedical science, we're often working with average effects, right? We try to look at, on average, what is the size of an effect? And I think when you remember the plots that I showed you here, I pointed always to the average effect. But there's a lot of individual variability in the exposure and in the response to light. Generally, this is sort of a wisdom that I think we know now, and I think it relates to the initial question or the initial message that I had, which is a healthy pattern of light exposure includes a rhythm of bright light and darkness every day. We can measure this, and we can ask to what extent are we meeting our recommended light levels. But there's things that modulate the effect of light. And so you remember the plot or the video of the pupil. Here what I'm showing you as a function of age, the pupil size. It's missing an axis here, but this is pupil size as a function of age. What you see is the older you get, the smaller your pupil is. This means that there's less light reaching your retina just because of age. It's a phenomenon called senile meiosis. It's very well known in optometrists and ophthalmologists because when your pupil size is small, it's very hard to look at the back of the eye. So if you want to do an eye examination in older folks, usually you need to use a dilating drop to make the pupils go big. And so age can influence the physiological effect of light in humans as less light reaches the retina because of aging. There's also substantial individual differences in the physiological responses to light. Here I'm showing you data that we collected in the laboratory where we are comparing a dark condition to a bright condition and looking at how much melatonin is produced throughout the entire evening. If it was the same for everyone, you would have basically a very tight clustering here. What you see is there's enormous variability in the total amount of melatonin that is produced by individuals. There's a group of people that are sometimes called high secretors because they secrete a lot of melatonin. This is also changing with age, so the older you get, the less melatonin you produce. But then when we're looking at the effect of light in terms of how it suppresses melatonin, we also see enormous variability there. So for some people, you almost have full suppression of melatonin, so the same light can almost fully block the production of melatonin, and then for some people, there's almost no effect. So physiologically, this is super interesting, right? Because it shows there are individual differences. We don't fully understand why they are so pronounced. We have data that show that the most sensitive individual is 30 times more sensitive than the least sensitive individual. So these are massive differences, right? Almost a factor of 30 is something that is really hard to find in other aspects of biology. And so we don't really fully understand what drives these differences. Is it genetic background? We know age has an effect, but if you limit the age range, you still see variability. And so these substantial differences ultimately are interesting from a biomedical point of view or biological point of view, but they also mean that when we are trying to evaluate the effect of light in the built environment, we cannot just assume that the effect is the same for everyone. In this group of, I'm very bad at estimating numbers, but I'm guessing 70 people, you will have some people who are very sensitive to light and some people who are not very sensitive to light. And I think this directly translates to the exposures that you might get. If you don't like light, you're not going to go outside to seek light. If you are a person who needs a lot more light than someone else, your light exposure will be different. And so ultimately, we believe that the sensitivity differences that we see are related to how much exposure we might get, and that's something that we'd like to test. Here's some data or some results that we recently compiled, at least in preliminary terms, which is that light exposures, the exposure you get varies by very simple questions, very simple factors. And so in our data set where we have more than 150 participants, including many from Costa Rica, we asked the question whether today is a work day or is a free day, and depending on whether it's a work day or a free day, you have different amount of light exposure. And surprisingly, during work days, you have more light exposure than during free days. We had a little bit of a discussion about this earlier, and so you can try to figure out why it is that you have these systematic differences. There's also differences in light exposure depending on sex. So if you're female, you on average have lower light exposure than males. This is a finding that is also was replicating a finding from a US study. And then being at home, intuitively you would expect this. If you're at home, you're not outside. And so as a consequence, if you're not at home, you have a lot more light exposure than if you're at home. But these are very simple drivers, right, of how much light exposure an individual might be getting that we can see in this data set already. So this brings me to sort of a wider question, and that is one where I'd like to reflect a little bit on how we do science. Before that, let's just recap. We've seen a lot about the sensory pathways, how light affects us beyond vision. We've seen effects of light on the circadian clock. We've talked a lot about light exposure and how it differs, including the light exposure you might get in places like Costa Rica. And we've talked about inter-individual differences, such as age, gender, sex, and other factors that influence the effects of light. These are photos that are representative of the types of study that we do in my laboratory, in my group. We have very well-controlled light exposures. We are interested in what does the light do to the retina. We have these intrinsic apparatuses that are basically developed to run studies to understand biological capacity. So what we do a lot is try to tickle out how can we stimulate a mechanism in a way to uncover a biological effect. Overall, we can say that the majority of studies on the physiological effects of light have been performed in the lab. Now, hopefully none of us lives in the lab, right? So there's a need to understand light exposure and the effects in the real world in a broad range of population, and this includes, of course, a perspective that recognizes that a lot of biomedical research comes from Western Europe or North America. And then ultimately, and I think this is to me a very important thing, which is this is an active area of investigation, and there's a lot still to be done to really understand how variability in exposure, how variability in response ultimately leads to effects on health and well-being. And so I want to close with the first thing that I started with, and that's this little mantra, this little rule. You should try to have bright days, you should try to have dark nights. So simple rule for circadian health: more light by day and less light by night. And with that, I want to thank you for your attention. Happy to answer any questions, big or small.
So what you said about light sensitivity, someone with higher light sensitivity would have higher melatonin suppression or not?
So it depends on how you define light sensitivity. So there's no one light sensitivity, right? So you can define light sensitivity as me asking you, relative to the population, would you consider yourself more or less sensitive? That's one way. I could also invite you to the lab. We could measure melatonin suppression and determine, relative to the existing sample, are you suppressing more or less? Unfortunately, we don't have a full understanding of whether your subjective evaluation of light sensitivity maps to sort of the objective biomarker-based measures. So that's something, of course, that we're interested in doing, but of course you need a lot of time to collect these data. So far, I don't know that there is, so it would be great, right, if I could just ask a question of, hey, how light sensitive are you? And that's predictive of how much melatonin will be suppressed, but we're not there yet because it takes a long time to collect these data. It's very expensive to run the biochemical assays. Participants come to us for four nights across 4 weeks, so it's just very resource intensive. But ultimately, we'd hope that we have sort of an understanding of, is your subjective understanding of how light sensitive you are mapping to objective markers? Yeah, so it's a really great question. I think of course, the approach that we're running with here is one where we kind of isolate one thing and try to really understand when everything else is constant and we vary that one thing, what happens. So usually in our experiments, we try to keep temperature constant because we don't want to have the variation induced by temperature. In practice, we know a few things of what heat and thermal factors do to sleep and circadian rhythms. We know sleep is a lot worse under heat when it's hot temperatures. I think everyone can attest to this. We also have interesting data that shows that if you put, so this is in fruit flies, right, which is a common model in biology. You have fruit flies and you have a rhythm of temperature. Their circadian clock will also entrain to temperature variations. So in a constant light condition, you're just varying the temperature, their clock will still run based on the temperature variations. One of the things that we're interested in with our big data set of now 191 participants across the globe is to kind of relate outside ambient temperature to one, how is it predictive of exposure that you get, but also how does it explain effects on sleep. So ultimately it's an empirical question of can we detect this in the data set we have. So that's outstanding. We're working on this. And so Andrea will hopefully be doing an analysis relatively soon, looking at exactly weather and how things like temperature, but also precipitation, rain, changes the exposure you get. And I think from a climate change perspective, there's another area that is, of course, implied or implicated, and that is the modulation of behavior, right? So the environmental factor of temperature is one, but of course it also will change what I can do and what I do and when I do it and if I go outside and how long I go outside. And so there is probably a direct effect of temperature on physiology, but there's also one that's behaviorally mediated, just because the repertoire of things that you can do when it's very hot will be very different. That's sort of, I think, I mean, in short, there's a lot to be done, to be honest. And I think with climate change, it's something that, of course, we need to take seriously, because we know that temperatures are gonna rise. And as a consequence, how do we ensure sleep and circadian health in this changing world?
Well, I have a question just out of curiosity. So you first saw the data and the results of Costa Rican people, and now how has your experience being here like changed your interpretation of what you had previously seen in the data?
I will say that I'm surprised by the amount of rain and the low temperatures that I've experienced over the last couple of days. So, so I think, I'll be honest with you, I had spent some time in Singapore, which is also in Malaysia, which is close to the equator. I had spent some time in Ghana, which is of course also close to the equator. And I was just expecting a type of heat that I usually don't encounter in Germany. I do think that my own experience is that, you know, you want to, I mean, the few days of sun or the few hours of sun that I've had, I tried to avoid it. And the days of rain, I also didn't want to be outside, right? So I think it's I feel like the lived experience corresponds to, I think, the data that we currently have. Yeah. That said, I think it's been a great visit. So I'm very happy to be here. Thank you. Great question. So the first one is iris color. So different people have different iris colors, ranging from light blue to very dark brown, right? And I think that's, you know, you can look in someone's eyes and kind of see where they are. We did a study where we tried to look at, does pupil size depend on iris color? And we didn't see any difference. I don't think that, I mean, I think from first principles you would expect that if less light is reflected by the eye, then more light goes into the eye, right? So if you're light blue versus a brown color, you should, from physics, believe that more light goes in. So far, we have not seen any relationship between iris color and melatonin. There are sort of these anecdotal reports that people with blue eyes are more light sensitive. I don't know that there's any robust evidence for that. And I think part of this is related to the question that you asked, which is how do you define light sensitivity and how do you measure it? Second question, so the IPRGCs follow the distribution of retinal ganglion cells. And so when we're looking at the retina, we have a lot of cones in the center of the retina. This is where our vision is sharpest, and it drops off as a function of distance from the center of the retina. The rods are different. They actually peak off the center of the retina, which is why when you're in dim conditions, you're trying to see something, you see it better when you're not directly looking at it. So if you look at a night sky, sometimes, and you want to see a star, if you don't look at the star, you will see the star, but if you look at the star, you don't see it. IPRGCs peak not in the center, but a little bit off, so roughly, let's say 8 to 15 degrees perifoveally. It just follows the distribution, the topographic distribution of retinal ganglion cells. There appear to be differences between upper and lower hemisphere. There's also some data that show that, how is it? So I think light from above, which is projected on the inferior retina, versus light from below, which is projected on the superior retina, will be more effective in melatonin suppression. This has been found, right? So light from above will be, at the same intensity, have more of an effect than light from below. You see these interesting differences, I think, in lots of, so, of course, why is this? Well, maybe the sky is always, in the upper visual field versus the lower visual field, but, it's hard to, I think, say why that might be. But it looks like, whether you look at superior or inferior retina, you have differences in sensitivity. But it hasn't been fully studied. Yeah.
Thank you so much for your time and for being here. Wrap up. So, muchas gracias.
Thank you.